Hello students, today we will be continuing with the chapter Sexual Reproduction Flowering Plants. Let us see about the pollination. Flowers are mainly responsible for bringing about fertilization and seed formation. To facilitate fertilization, it is necessary that the male gametes inside the pollen grains and the female gamete inside the ovule are brought together. However, since the gametes are motile, pollen grains have to be transferred to the stigma. This process of transfer of pollen to the stigma of a flower is brought about by agents such as insects, wind and water. It is called pollination. Pollination can be of two types: self-pollination and cross-pollination. When pollination takes place within the same flower or between flowers of the same plant, it is known as self-pollination. In plants, self-pollination can occur either through autogamy or geitonogamy. In autogamy, pollen is transferred from the anther to the stigma of the same flower, and it is seen in cleistogamous flowers. Cleistogamous flowers means the flowers which do not open at all, and these are produced by plants such as Viola oxalis, Commonelina, Arachis hypogea, or it is also called as peanut. The anthers and stigma of these flowers lie very close to each other, and there is a dehisce of grains, and it gets deposited on the stigma, which ensures the production of seed. And it sets even in the absence of pollinators. Autogamy, however, is rare in chasmogamous flowers which open and expose to their anthers and stigma. It requires a synchrony between pollen release and stigma respectively. Now next is geitonogamy. In geitonogamy, the second type of self-pollination, pollen is transferred from the anther of one flower to the stigma of the another flower of the same plant. Apart from self-pollination, cross-pollination has also been commonly seen in plants. Type of pollination involves the transfer of pollen grains from the flower of one plant to the stigma of the flower of the another plant of the same type, and this type of pollination is called xenogamy or allogamy. Interestingly, xenogamy is the only type of pollination that brings genetically different type of pollen grains to the stigma. So in both cross-pollination It's a cross pollination taking place. So from one of the anther, it has been carried to the stigma of the another plant. Only one type of pollination can take place, which brings genetically different type of pollen grains to the stigma. So in both cross-pollination and self-pollination, pollinating agents play a vital role. These agents include biotic agents such as birds, bees, butterflies, bats. So these are the mammals, bats are the mammals. And abiotic agents like wind and water. Usually, pollination takes place by biotic agents which get attracted by flowers which vary from each other to attract their specific pollinator. Insect pollinated flowers, for instance, are usually large and fragments. However, if they are small, they bunch together in the inflorescence to make them conspicuous. These flowers produce pollen grains as well as large quantities of nectar to reward the agents. When pollinating agents come in contact with its body, it is covered in a coat of sticky pollen grains. So, when the agent sits on the another flower to suck the nectar, the pollen present on their bodies comes into contact with the stigma, thus completing the process of pollination. So you can see the pollination in the major agents. They are coming and sitting on the nectar. Conversely, flowers pollinated by butterflies are usually lightly scented and brightly colored with nectar, not very deeply hidden, while those pollinated by the beetles and flies, they secrete a rotten odor. Flowers pollinated by birds, on the other hand, have large odorless orange or red tubes which are rich in honey. Those nectar will be having more honey. And certain plants attract pollinating insects by offering them shelter. Instance, Amorphophorus. It's a 6-foot tall flower. It attracts pollinating insects by offering them a safe place to lay their eggs. In the yucca plant, on the other hand, a female moth, it transfers the pollen of the another flower, to the stigma and simultaneously deposits her eggs in the locule. You can see them. The flowers ovary with larvae which hatch out of the eggs start consuming some of the developing seeds while leaving enough of them to propagate. So both work is taking place. So the plant does. Both the moth and the plant are dependent on each other for their completion of their respective life cycle. So both development is taking place. The pollination as well as the seed formation. However, certain birds and insects such as bumblebee visit flowers for nectar and pollen, but they don't bring about pollination. These are referred to as pollen robbers. Honeybees as well as other insects pollinate a large number of aquatic flowering plants such as freshwater lily. However, in certain freshwater plants such as Vallisneria and Hydrilla and marine seagrasses, you can see them. The pollination, it occurs in the water. So this is known as hydra freely. In Vallisneria, for example, the female plant produces long stalked female and flower that reaches the surface of the water. The main plant, on the other hand, remains submerged in water, where it produces several tiny floral buds. You can see them. Upon maturing the buds, they arise to the water surface. See, they go in, they attach to, so it's arising to water surface and open up to expose the anthers. Pollen released from the anthers is scattered by water currents, where some of them eventually come in contact with the stigma. The male and female flowers remain submerged in water. The male flowers produce longer and ribbon-like pollen grains which have a mucilaginous covering that protects them from getting wet. The pollen released in water is carried by water currents towards the submerged stigma, and thus pollination is completed. Surprisingly, in aquatic plants, wind pollination, also called the nemophily, is more widespread than water pollination. So these are all wind pollination also occurs in terrestrial plants. It is grass, bamboo, coconut and maize. And these plants possess a compact inflorescence, male inflorescence and female inflorescence, with well exposed stamens that allow easily dispersal of pollen, and large feather pistil, which makes it easy to trap the pollen. The pollen, which is light and non-sticky, it is produced in large quantities. A single flower of cannabis, for instance, produces 5 lakh pollen grains to compensate the loss of pollen associated with wind pollination. So thus wind, water, mainly takes part in pollination as major agents, as well as biotic pollinating agents also plays a major role. This is an important process which ultimately leads to the fertilization and production of seeds in plants. Production of flowering plants pollination is a mechanism that aids in transferring the pollen released from the anther to the stigma of the flower. If this pollen is of the right type, the stigma induces it to germinate and produce the pollen tube. In the pollen tube, it is containing the 2 male gametes. You have studied in previous class. No? Gametes then secretes hydrolytic enzymes which help the pollen tube to digest the tissues of the style as it travels through, and it enters to the ovule through the micropyle below the ovule, and inside the ovule the pollen tube encounters the embryo sac. See how it of the female gametophyte containing the two synergids. The synergids and the egg cell and a central cell with the two polar nuclei. Now here the pollen tube now enters one of the synergids through the filiform apparatus, during which it ruptures and releases the male gametes. The male gametes into the synergids with the cytoplasm. That's how it is entering. See the two male gametes. One end is for the egg cell and fuses with its nucleus. This fusion is known as syngamy, or the first fertilization, which results in the formation of diploid zygote. So meanwhile, the other male gamete, it moves towards the two polar nuclei and fertilizes them. It is called triploid primary endosperm nucleus or primary endosperm nucleus. This is known as triple fusion, as it involves the fusion of three haploid nuclei. Now after the triple fusion, the central cell is called the primary endosperm cell. So did you know that syngamy and triple fusion together they are called as double fertilization? The phenomenon of unique of flowering plants. Double fertilization is followed by several post-fertilization events which eventually transform the ovule into the seed and the ovary into a fruit. Each part develops to the corresponding cells. Okay. In these events, it includes degeneration of the antipodal cells, synergids, and the development of the zygote into an embryo. And the primary endosperm cell into the endosperm by repeated mitotic division. So students, each part, until now you didn't know exact how the fruit is been developed. Actually, from inside the flower you are having the ovary, and that keeps developing into the fruit. Each part, the layers of the ovule, that develops into a fruit. So types of endospermic development. Here you have nuclear endospermic development and cellular endospermic development and halobial endospermic development. See, cellular endospermic development and halobial endospermic development. The nuclear endosperm development is the most common type of endosperm development. That is, from the primary endosperm nucleus divides mitotically and forms the free nuclei. So mitotic division takes place. This division sometimes followed by the cytoplasmic divisions from the periphery, from the sites. In case of the cellular endospermic development is seen in plants such as in utricle area. The division of the primary endosperm nucleus is immediately followed by the cytoplasmic divisions. In halobelic endoplasmic development, the intermediate type of the development, where half of the cells develop in cellular mode and the half undergo free nuclear division. Interestingly, the number of free nuclei formed prior to the post-fertilization differs greatly. In case of tender coconut fruit, the water is nothing but the few nuclei. There are thousands of nuclei there. As the coconut matures, The cellular fertilization leads to decrease in the quantity of water and the formation of white kernel, the cellular endosperm. So therefore, in the coconut, we find both free endonuclear endosperm made up of thousands of nuclei and cellular endosperm. Now we can see both free nuclei and cellular endosperm. Histological studies have shown that the endosperm contains stores food in the form of starch or proteins as in cereals or oil as in castor or coconut. So this food provides the nourishment to the zygote. It is developing into the embryo, and it is interesting to note that the endosperm also forms a major source of nutrition for human beings. Incidentally, the development of the embryo, also known as embryo gene, because only after the certain amount of the endosperm has developed, you can see these events. So in fact, some plants such as groundnut, they belong to the bean family. The mature seeds lack endosperm as it gets completely absorbed by the developing embryo. When these seeds germinate, the cotyledons or seed leaves become the source of nutrition for the embryo. However, in the mature seeds of castor, we see that the endosperm is retained as a storage tissue. In case of cereals, the allurin layer, which forms a part of the sperm. The sperm stores the food as well as secretes the amylase enzyme for the digestion of the stored starch during the germination of the seed. Studies by botanists have revealed that during the early embryo gene, the zygote divides into two cells. The basal suspense cell towards the micropylar and the terminal embryo cell towards the chalazal end. The basal suspensor cell further undergoes mitotic division and they form 4 to 8 cells which push the terminal embryo cell into the tissue of the endosperm to allow it to derive nourishment. The suspensor, now you can see. The temporal embryonal cell also undergoes divisions to form a group of cells known as proembryo, which subsequently develops into a globular, heart-shaped and torpedo-shaped mature embryo. Dicotyledonous embryo is nonetheless different from that of the monocotyledonous embryo and the dicotyledonous embryo, for instance. The embryonal axis in the two cotyledons, the epicotyle, the portion of the embryonal axis above the level of the cotyledons. With a plumule, you will there at the top, below the level of the cotyledon, is a cylindrical portion called the hypocotyle that terminates at the radical root tip, which is covered in the root cap. On the other hand, the monocotyledonous embryo are commonly found in grass family and has only one cotyledon called the sputum situated towards the lateral side of the embryonal axis. The upper portion of axis is known as the epicotyle, which lies above the attachment of the scutum. The epicotyle has a shoot apex and a few leaf primordia enclosed in the coleoptile, a hollow fuller structure. The lower end of the embryo axis, the radical and the root cap covered in the undifferentiated sheath, is called a coelorhiza. So various parts of the embryo, they are developed into different seeds germinate. In this manner, the embryo as well as the endosperm develops from the zygote, and the primary endosperm cell after fertilization has taken place in flowering plants. Fruit means mango, papaya, apple or banana. However, for a botanist, the fleshy and the juicy mangoes or papayas are much as a fruit as our groundnuts or cashew nuts whose out dry in nature. This is a long section of ovary in papaya. From the ovary post-fertilization, incidentally in most cases, fertilization, the development of the fruit triggers with the withering of the parts of a flower including the sepals and petals and also the stamens. However, in some cases like apple, floral parts such as thalamus become the part of the fruit, which other parts of the flower besides the ovary are involved in the formation of the fruit and is called false fruit. And the other fruits like mango and papaya that develop from the ovary are called true fruit. While mango and papaya developed after fertilization, fruits like banana developed without fertilization and are called parthenocarpic fruit. Moreover, in the case of banana, the edible parts developed from the inner tissues of the ovary, and the brown specks that we see are the remnants of the ovules. If we cut open a wood that is made up of two parts, and berry cup and one more seeds. The pericarp is a fruit wall that develops from the long walls of ovary. And the seed, the fruit, actually these are fertilized. And typically a seed is enclosed in a tough outer protective covering called the seed coat, which is usually hardened integument of the ovule. These integuments develop into the outer part. The pile of the ovule persists in the seed is a small pore through water which oxygen can enter. So inside the seed coat there lie one more cotyledons as well as endosperm embryo and embryo neural axis. The cotyledons, which are often fleshy and swollen, contain abundant food reserves. In the endosperm, the other hand, is a nutritive tissue nourishing the developing embryo. Interestingly, in certain plants such as bee and pea, the developing embryo completely consumes the endosperm. Such seeds that contain no endosome are called non-albumin seeds. However, wheat and maize residual of endosperm is seen as it is not completely consumed by the embryo during the developmental stage. Such seeds are called albumin seeds. Sometimes in addition to gender endosperm, seeds also contains remnants of new cells called the pensum. Stores food material. Such seeds are called as perispermic perisperm that also stores food materials like black pepper is common example for perisperm. Next, but there is a firm link on albumin seeds. As the embryo begins to develop the seed, it starts to mature and lose its water content to become relatively dry. The mature seed therefore has only 15 to 20% of its mass as moisture. Moreover, the embryo's growth rates slows, and under unfavorable condition, it may enter a state of inactivity called dormancy, in which the embryo develops an increased resistance to desiccation. Conversely, conducive conditions that provide favorable temperature and appropriate water awaken the dormant embryo, and the seed germinates with the embryo developing into seedling. This ability of seed to germinate is called seed viability, and it is called seed viability. It varies across the seeds. The seeds of spinach remain viable only for a year, whereas muskmelon seeds, they remain viable for 5 years. So in Arctic region, they have led to finding of seeds of new perisperm which remain viable for 10,000 years before germinating and flowering seeds. A character is unique to angiosperms. They also offer many advantages to the groups of plant seeds. They have special mechanisms such as bursting of wings that help them in disperse to new habitats, and thereby allowing these plants to climb to new areas. In addition to the seed's tough outer coat protects the embryo from hostile environmental conditions. And these have better survival rate as guaranteed nourishment by the cotyledons. As seeds are products, they help to bring about the genetic variations within the plant species. Genetic variations are the key to evolution, and also they help the plant species adapt and survive in different environments. Seeds which help in the propagation of the angiosperms. So from the agriculture, interesting, the farmers in the recent times have started using hybrid seeds developed through artificial cross-pollination techniques. These seeds guarantee increased the productivity and superior crop quality. However, when the seeds are borne by hybrid crops, they are sown. Seedlings do not exhibit the hybrid characters to the new segregation. This forces farmers to purchase new hybrid seeds year after a year, which is an expensive affair. To address this problem, botanists are trying to understand the genetics of the atomics as and transfer the genes into hybrid plants. That is, the apomixis is a form of asexual reproduction in which the seeds and embryos are formed without fertilization. In fact, the seeds or the embryos either developed from the unfertilized egg, which do not undergo meiosis, from the cells of the integument. And the sellers of the botanists believe that converting hybrid crops into permits will prevent the segregation of characters and seeds produced by the hybrid crops, and thereby eliminate the seeds need to purchase the hybrid seeds. And every year, as the seedlings produced by it permits they resemble their parents genetically. So no segregation character. Plants such as grasses and those belonging to the family Asteraceae and Rutaceae are the common example of community. Moreover, the certain such as citrus jambre, which belongs to blue Jesse family, the new cells surrounding the embryo sac, they will divide, protrude into the embryo sac. Later, these cells develop into embryos, and hence a single seed contains more than one embryo. This phenomenon of having more than one embryo in the seed is called poly. So in this session, I have given only 3 questions, though I have covered little larger portion. In next class, I will be sharing you still other questions. Okay. So here I explain the types of pollination, that's the 3 types, and define anemophily or ornithophily. Though these 2 are often repeated questions, I have given you this. And explain double fertilization. Remaining dicotyledonous seed, monocot, they are also important. I will be giving them in the next class. Thank you.